which schools are having exams? Some schools are having exams. Let's see. How many? One. Both are the question. Both are the side. Are we done with asexual modes we had started? Check the book. Check your books. Asexual modes of reproduction we must have started the other day. Which methods are we done? Check. Spore formation. Okay. Yes. Yeah. We are done with binary and multiple fission. We are done with fragmentation. We are done with budding. Budding done. And regeneration, did we talk about? Regeneration we haven't discussed, and spore formation. Let us complete first with spore formation. Asexual reproduction, still a few more topics. So, first that we talk about asexual reproduction. For asexual reproduction, let us first understand spore formation. Spore formation, what type of organisms show this method? So for spore formation, the type of organisms that show this method, you'll have to remember. What type of examples will show this process of इसमें चार और इसमें पूरा खाली देखो इधर आ जाओ एक दो. Now, what type of organisms will show this method? Spore formation, majorly fungi. You'll have to remember some examples of fungi, such as to name some of these fungi, which are some examples: rhizopus, then the mold, then the mushroom. So these type of examples you'll have to remember. So what type of organisms carry out spore formation? All these different types of fungi. The mold that we understand, the mold majorly found in different type of food items. You must have come across bread mold. Kabhi bread mold dekha? Bread mold, did you ever see? Bread mold, when we are talking about the process of spore formation, how do they multiply? They always multiply by this process. Bread molds that we understand, this would be something this way. This is how this type of organisms look like, the process of spore formation. So we will sit straight, huh? No looking back, straight on the board. Now, when we are talking about the concept of spore formation, this is the method. The bread, any type of bakery items you must have come across, all of you. Molds on the citrus fruits, on the surface of the citrus fruits, some green, black patches that develop when the fruit is overripe. Molds again. Mushroom, or well known already, you know what type of organisms where they are found. Rhizopus, where rhizopus, these are type of fungi which we come across in, say suppose now that it is monsoon time, we get to see them on the surface of the wood, surface of the leather goods. If you talk about your shoes, your bags which we are not using in this month of monsoon, take them out from the cupboard, you get to see a white layer develop on them with a typical smell also and odor also. Wooden cupboards, a number of times, if they are not been open for a number of days, they develop a white, granular layer. So what type of fungus getting developed there because of the moist weather? This is rhizopus. Now how do they develop? These are multicellular organisms. These type of organisms, they have got simple multicellular organization, and this simple multicellular body will start developing from the spores. These are multicellular structures. What are these? Known as fine filamentous structures, which they develop first of all. These are known as hypha. The filamentous structures known as the hypha. And then one of these cells, they will start showing multiple nuclear fission, quite similar like those multiple fission that we had come across. So one of these cells, now they will start showing multiple nuclear fission this way. This is the multicellular structure, the hypha that I mentioned. And then this is followed by nuclear multiple fission. Nuclear division multiple number of times, resulting in the formation of nuclei first of all. But now that these nuclei mature, they are also known as spores. So spores is a word that you have to remember. Spores, multiple nuclei formed from the division of the nucleus in one of those cells, resulting in the formation of the spores. But now these spores, quite a number of times when these spores are too many, I talk about that these spores, when they are now too many, what will happen? These spores, they will start getting released from this type of swollen structures. And what are these swollen structures now known as? These swollen structures now are also known as the knob-like structures, are also known as the sporangium. These words to remember. So what is this knob-like structure known as? The knob-like structure known as the sporangium. What is this filament now known as? Earlier when the spore was not getting formed, we had named it as hypha. But now when the spores are getting formed and a large number of spores getting released by the breaking down of their wall, now we will term them as the sporangiophore. The sporangiophore is now the filament, giving rise to a knob-like structure at the tip, and this knob-like structure containing numerous spores. What is it known as? Sporangium. And the fine granular structures which are released, these are known as the spores now. How is it different from multiple fission? It is different from multiple fission in various aspects. Here we are talking about process of spore formation taking place in a multicellular organism. In case of multiple fission, what was the example we studied where multiple fission takes place? In which organism we had studied multiple fission taking place? What type of an organism was it? Multicellular, unicellular, what was it?
Unicellular.
Unicellular, that was. Okay, tell me what was the name of that organism we had studied? Multiple fission is in Plasmodium. Multiple fission is always in Plasmodium. It's a parasite. So now, how is it different from multiple fission? We are saying that yes, the nucleus divides into a number of times. Multiple fission in single cell organism. Spore formation in multicellular organism. And now that these spores are released, these spores, they may remain in the air or on any particular surface. They would not start growing, but they may remain alive, inactive for a long period of time. How? Because these spores, they start developing a protective resistant cyst wall, a covering known as the cyst wall. So this cyst wall will not allow the spore die out, even if the environmental conditions are not favorable for their growth and maturation. So this particular cyst wall allows the spores remain active for a long time. They may not grow, but they will grow only when, who has the sheet? So they will grow only when the conditions are suitable for their process of growth. So now, when we are talking about the process of spore formation, the methods to remember, the differences to remember. The next method of asexual reproduction. Asexual reproduction, these are the actual methods of asexual reproduction now that we are going to talk about, that is regeneration. We talk about regeneration in different organisms. Regeneration, what is the definition of regeneration? Process by which Process by which an organism can regain its lost parts. Organism can regain its lost parts. How? Its lost parts can be regained only by the process of simple cell division. By the process of simple cell division, it can regain its lost parts. But now, when we are talking about the concept of regeneration, the concept of regeneration in various group of organisms, I may talk about the process of regeneration in lizards. I may talk about the process of regeneration in planaria. Let us talk about regeneration in planaria. How is the regeneration in planaria different from the regeneration in the, for example, I talk about regeneration in planaria different in the regeneration in the vertebrates. Now, this is a typical example that you are expected to draw as per your syllabus. This organism, planaria, present in the shallow waters. Now, this type of an organism, the planaria, it's a flatworm. Not a disease-causing organism, but though we are talking about the flatworms, it does not cause any harm to humans. But now that this flatworm, accidentally when its body gets divided into 2-3 segments, accidentally body gets divided into 2-3 segments, what will happen? If it is the lower body lost, the upper body will simply undergo cell division, form the lower body. The lower segment having quite a number of cells, adequate amount of cells are there, regenerative cells are there. Then this lower body also can undergo cell division from the upper body. क्या हुआ?
अभी उसका डेथ होगा ही नहीं.
किसका नहीं डेथ होगा?
प्लेनेरिया.
हाँ तो डेथ क्यों होना है? The body has divided into 2-3 parts. The concept is not we are talking about death. We are not talking about immortality here. Here we are saying that this is an organism. Is it normally, naturally dividing by this method? No, this is not a normal method of division. This organism, I just now mentioned that it is present where in the water body. So by chance, sometimes what happens, it is eaten by its predator. It is eaten by its predator. So that means part of its body is lost. So the upper part, if eaten away, the lower part can regenerate from the upper part. So another new organism is developing. If the body, now this is an aquatic organism present in the water body, quite a number of times it moves through the hard surfaces at the surface of the water body between the rocks and all. So what happens quite a number of times? Body divides into 3-4 pieces also. If the body is divided into 3-4 pieces, are we getting 4 planaria every time in such situation? Not necessary. How many regenerative cells are there based on that? I may talk about a planaria with this respect. I may talk about a planaria where this organism has the entire body of that organism divided into a few parts like this. So I'm saying that will this part also give rise to a new organism? No, the number of regenerative cells very less here. So when you write your answers, this concept you'll have to write that how many organisms will this single organism, or is there any standard number like fission? Binary fission, parent will give rise to 2 organisms always. So essay regeneration, is there any constant number? Nothing. How many segments or how many pieces the entire parent organism is divided into? Maybe this part also will not give rise to a new organism. This may give rise to 2 new organisms. This will give rise to the middle and the lower part. This will give rise to the upper and the lower part. Definitely here we are saying that from one organism, 2 organisms we are getting. At times, one organism, 4 organisms we are getting. But this is accidentally when the body is divided into parts. By simple cell division, they can form the upper, lower part and complete the organism. Is this the natural method of reproduction for them? No, that is not the natural method of reproduction for them. What is the definition? Always you should remember for regeneration, process by which an organism regain its lost parts by cell division. तो ये plenary के लिए तो हो सकता है. Can this happen for the lizards? Now I talk about a vertebrate. If I now talk about a vertebrate, in case of the regeneration for the reptiles, हो सकता है ऐसे? हो सकता है ऐसे? I talk about the reptiles. Now, these reptiles are in the process of regeneration. So, can I understand that this reptile, which is in the process of, this reptile has lost a small part of the tail, a small part of the limb. By following this rule, am I to understand that this tail also will give rise to a new reptile and this will also form the reptile name? That concept will not work here because it is a vertebrate. This is a simple invertebrate. No organs, no tissues, not much of systems, no systems present, no bones, no cartilage, कुछ भी नहीं है. So this organism by simple cell division can regain and complete the rest of the body and the new organism can happen. But here why not? Because this organism at the most can heal up this lost part of the body. Skin formation हो सकता है. But this will never be able to give rise to the bones and the cartilage and the systems and the organs to complete the organism. There's a question in your book there. Can complex organisms give rise to new organisms by regeneration? Answer is always no. Complex organ, give reason. क्यों नहीं? It is there. Check करो. Textbook देखो तो सही. Now, when we are talking about the concept of regeneration, it is before. It is before. See there. So, when we are talking about the concept of regeneration, now this is a typical example that is there in your book, the previous example that I. Earthworms also same thing. Earthworms also can regenerate and form the new organism because they don't have. Because they don't have important organs and systems, bones, tissues, कुछ भी नहीं है ना. So that's the reason.
उसमें जो दोनों जो पार्ट होंगे डिवाइड होंगे वो दोनों पार्ट ग्रो करेंगे. दोनों
for invertebrates they can grow and give rise to the new organism. So that is not to be considered as a method of reproduction. Why? Because that is not the regular method of reproduction. These organisms, they have got this type of organism, they reproduce by fission. So always to remember, I may talk about organisms. What are some organisms that undergo the process of regeneration? The first one that I'm talking about is planaria. I talk about organisms, annelids. Quite a number of annelids show the process of regeneration. Yes, these type of organisms accidentally, when their body divides into 2, 3, 4 parts, possible that by the method of cell division they can give rise to new organisms. Numbers, not anything standard, not any constant number that is there. But I may also talk about the regeneration of vertebrates like the lizards, regeneration of the amphibians. So in those cases, if the limb, a small portion of the limb is lost, what will happen? The body will just regain a small portion of the skin and the muscles. Quite a number of times the entire limb is not formed. We talk about even in human, when we come across people who has lost their finger or the toe or the limb, we have come across that these people had only been able to regain, the body has been only regain the skin has grown, and then the healing of the bone, the cartilage and the bones will never grow, because the entire process of growth for us starts, for all vertebrates, starts from the embryonic stages. There's a proper method that cannot develop at any time during our lifespan. So that's a concept always to write down. Lizards and regeneration here, how it is different. You get questions again in a different way. How is the method of regeneration different in lizards and in planaria? So you'll have to write down the invertebrate and the vertebrate concept. The complex organisms cannot give rise to new organisms from the cut out portion of the body, because in case of complex organisms, the development is all programmed right from the embryonic stage. The tissues and the organs and the bones and the cartilage, everything has to be developed stage by stage. Anytime during lifetime cannot happen. Here it is simple. Is that clear? This is not a complete proper method. This is not the regular method of reproduction, asexual reproduction. So how many methods of asexual reproduction we had studied? Fission, 2 types of fission, binary, multiple. Diagrams draw here. Did you all draw the diagrams? Both the diagrams very important. We had studied fragmentation. Fragmentation very important. Next is we have talked about budding. Budding in hydra, budding in yeast, budding in which plant? Bryophyllum. So that is the types of budding now to talk about. Next is, apart from that, what else we have studied today? Spore formation. And now regeneration. So how many diagrams? Fission ke 2 diagrams, fragmentation ke 1, and then followed by spore formation, budding in hydra 6, and then this one. So all these diagrams are very, very important. You please note it down in your notebook. Some of you had drawn the diagram, some of you had not drawn. The other day I had drawn this, I had shown you these diagrams. Fragmentation. Sponges, non-living. Sponges are the organisms which are present in the water. These are poriferans, these are living organisms. So this is not the sponge. That's not that sponge. The sponges are organisms. These are present in the water. The porifera, these are known as sponges because their body pura porous rehta hai, enough amount of water filled in. So these are organisms which can regenerate also. Starfishes also they can regenerate. So all these starfishes, sponges, they all will come in this category. And the lizards and the amphibians, different category, vertebrates. Okay. So these diagrams, note down all these diagrams you'll have to practice. The budding in hydra, that's a very, very important diagram. Now one diagram from this chapter, 2 to 3 marks. Definitely every year one diagram from this part of the chapter is there. Labeling, proper labeling. The last day we had done about fission, we had done fragmentation, we did budding, and now this diagram all of you will have to draw and practice, and along with that. So keep one page, remember that these diagrams to complete. Now this is about asexual reproduction. Now we move on to another concept of asexual methods of reproduction, partly asexual methods of reproduction, but in plants. What is that known as? That is known as vegetative reproduction, vegetative propagation. Let us learn what is vegetative propagation. Now talking about another asexual method, we say that is still asexual method because we are not talking about formation of reproductive cells yet. So another concept of asexual reproduction is vegetative propagation. Propagation, a simple meaning, to multiply, to grow in large numbers. Vegetative, what is the meaning of vegetative? The non-reproductive parts of a plant. Which are the non-reproductive parts of a plant? The non-reproductive parts of a plant would be the non-reproductive parts of a plant. What are those? The non-reproductive parts are the stem and root and leaf. So the stem, the root, the leaf, the buds, these are all the vegetative parts of a plant. Let us learn some methods by which the process of vegetative reproduction takes place, vegetative propagation takes place. Planaria, earthworm, starfishes. Starfish bhi hai. So now when we are talking about the concept of vegetative propagation, let us understand the process of 3 major methods of vegetative propagation we are talking about. Let us learn those specific methods. Now, cutting, a very simple method. Try to understand. The first method that we talk about is cutting. Cutting, very simple to understand, where we are coming across that in this process, majorly the root or the stem is used. Which parts are used in the method cutting? Generally the stem or the root. These are the 2 major types of plant parts which are used. What is happening? From a parent plant, certain length of the root stem are cut out. What precaution to remember when we are talking about cutting out? The cut has to be in a slant like this. The cut has to be in a slant like this. Why? The cut has to be in a slant like this. We cannot cut the stem straight. If you are cutting the stem straight and we put it in the soil, probably most of the time it will not grow. Do you know what the reason to write in your answer? Why the cut has to be slant? Quite a number of times you must have seen the gardeners and all how they slash it off in this, spiny and sharp, and then they put it immediately within 2-3 weeks. Roots will develop. So what is the answer to write down? Why cut it this way? Cut it this way for large surface area. Large surface area for what? Large surface area for the absorption of water from the soil. Large surface area for the absorption of water from the soil. So now in this way, when we are cutting the stem and putting in the soil, there is maximum probability that the stem will not die out, and roots will start now developing in a period of 2 to 3 weeks. Please come. Mere sir? No. Not me. Did you say? Now, when we are talking about this, that this surface, the large surface area, when we are talking about the large surface area, always to write why the cut has to be always in a slant, why the cut in a oblique manner, why the cut oblique. This is what the answer that you should write, large surface area. Otherwise, when we cut it, by the time the stem establishes, starts absorbing water, the upper part of the stem will all dry out. Yeah. So now this is the only precaution when we talk about cutting. So by this, what is the minimum length? 3 to 5 centimeter. So one particular plant can give rise to so many such pieces of the stem. Put them in the soil, new plants develop. What will be the new plants like? Will they be all identical to their parent plant, or yes, different plants will have different characteristics? Same. So in case of cutting, what are we to understand is plants are all identical. We are talking about vegetative propagation. The first method, plants are all identical. So there are quite a number of fruit plants. There are quite a number of flowering plants. Majorly flowering plants are grown by this. Few fruit plants which don't grow from the seed flower, they are also grown, but majorly flowering plants are grown by this method. The stem, root cut into segments, put into the soil, new plants are developing, but this feature is always there. The next is, we talk about the next method is layering. The next method is layering. Now, what is layering? Did you ever come across the grasses in the soil? When you try pulling those grasses, एक दो ग्रास प्लांट्स को अगर हम खींचते हैं, did you ever observe that the surrounding के बाकी जो भी सारे plants है, that also start getting pulled? तो एक ग्रास प्लांट्स को निकालना मुश्किल है, because the others also will start getting pulled, no? What is it? Because they all remain connected by their roots. They all remain connected by their roots. Why do they remain? They all remain connected by their roots because this is a method of layering that we are talking about. Let us talk about some examples. Artificial and pura, they will develop. So now, what is the natural method of layering? Let us come across a natural method of layering. In this method, when we are talking about layering, this is an artificial method actually. This way, this is an artificial method. Quite a number of times we come across the grasses. What type of plants will generally show this method? Naturally? ये तो artificial method है. I'll talk about this also. Layering is done artificially also. Majority of these plants, now that I talk about the jasmine hai, grapes hai, strawberry hai, these type of plants which have got comparatively softer branches and stem. Branches not very rigid and strong. I cannot talk about this type of method for the mangoes and the guava because they have the branches very rigid. bend करना मुश्किल है, they'll get broken. But if I talk about plants like the jasmine and the lily, and then we talk about the strawberries and the grapes, these have got branches, as in when the branches develop, they are quite softer, flexible होता है. And then that's the reason quite a number of times they will all start growing through the soil, touching the entire surface of the soil. So when they are touching the surface of the soil, wherever they touch, roots will develop. Something like when we talk about the second method, that is layering. If I look into certain type of plants, maybe now I talk about some grass plants are there. So these grass plants, they have got their leaves. Now these grasses, they also will develop some roots this way. They will also develop some roots this way, which are running on either side. They are running on either side. Now that the roots touches the soil, here again, now the main roots develop. So these are actually known as the adventitious roots. जो secondary roots होते हैं from the stem running above the soil, इसको adventitious roots कहा जाता है. And then this will start developing its main root, and another plant has started developing here now. This is what is naturally happening. तो artificial कैसे किया जाता है? Artificial, like I just now mentioned for the grapes, strawberry, jasmine, all this, the farmer wants to grow large number of plants. So इसको क्या करेंगे? This type of plants, now that the plant has and branches are coming up, and when the branches are coming up, they will start bending this branch towards the soil because it is soft, easily doesn't break. So when these branches are growing, they will always try bending some of these branches towards the soil. But will it stay like this? No, they will not. It will not stay. It will start reverting back. So, इसीलिए इसको थोड़ा सा soil डाल के make it stiff. काफी a number of times what they do is they will tie it with a bamboo or a long stick dug inside ताकि support रहे, and then the branch will not come up. Something which is shown here. So in a number of times we come across that yes, this branch which is now still growing, and then एक ही branch से और एक ही root से a number of these type of structures can be seen. So from the same branch also a number of times they will do this type of methods, allow this branch to grow and manually touch them in the soil, and then support every time. They will use a stick bamboo support and then tie it up. In 2-3 weeks time, when this adventitious roots are pulled, it is found that they have already become firm with the soil. We pull them, no, they doesn't come up. Already formed, support with the soil, firm with the soil, meaning the roots have already developed now. So तभी इसको निकाला जाएगा. This particular bamboo and etc. will be now removed out, and it is only the root remaining. And it is found that in another few more weeks, a new plant has already developed. So one plant is giving rise to another few more plants. ये naturally भी हो रहा है, and this is artificially been also done. Layering can be natural. Layering is also done artificially. Now that one plant giving rise to another 50 plants, are the plants identical? Yes, all plants are identical. What is the similarity? Cutting and layering, plants are all identical. Large number of plants can be grown at a short period of time. The farmer need not worry about any environmental factor, etc., etc., because this is more or less a surer method. So now we talk about two methods. What are these? Cutting and layering. Second, the third method that we talk about is a little bit different now. We talk about the third method. What is the third method? The third method known as grafting. Grafting is 2-3 plants. It can be 2-3 plants. So now, when we are talking about a concept of grafting, in grafting, what is done? In grafting,
मैंगो का एक स्टिक को ऐसे 2-3
मैंगो या तो और भी बहुत सारे plants. I talk about the guava है, pineapple है, various different type of plants. What is that?
इसमें मैम ज़्यादा
इसमें
इंडोर प्लांट्स
no, All these different type of fruits now that you are consuming, most of the fruits that we consume nowadays are all improved varieties. The type of fruits that we eat nowadays, quite a number of these fruits, guava, all improved varieties are. That's the reason if you ask your parents, if you ask your grandparents, the type of guava that we get today and the type of guava 2 generations back, not the same at all. We don't get those classical guavas at all nowadays because these are improved for some reason or the other.
इतने बड़े-बड़े इसमें guavas.
हाँ, इतने बड़े. And then the pumpkin, and then the pineapple, bananas. Definitely, these are all hybrids. Apple. And custard apple, so many different type of fruits, grafted varieties. What we are eating nowadays are mostly all grafted, ya to hybridized. So when we are talking about grafting, are you able to understand that it is two different parts of the plant being brought? But ऐसे क्यों किया जाएगा? Why? For what good reason this particular method is being done? Let us try understanding. Simple.
That the plant
improved varieties. For example, I may talk about one type of a mango. This is one type of a mango, and in this type of mango, what has been observed is it has got various desirable characteristics. Something like the fruit is of a moderate size. Okay, not a problem at all. The fruit is of a moderate size. The fruit is nutritive also, high nutrient quantity. But then next is when we are talking about that this plant is very much sensitive to pests. What is the meaning of this? Easily it gets affected by different type of microbes in the soil. So the farmer is always facing this problem. Never mind, this type of fruit is nutritive, commercial demand bhi hai, everything is good. But still, by the time it is half of the season, it has started growing. This is how the plants get infected by fungus. This is how the plants get infected by virus. So the production as expected kabhi bhi farmer ko nahi mil raha. And then he also has been coming across that there is another type of a mango. In this type of a mango, what is there? In this type of a mango, the fruit size is maybe smaller. So that's the reason commercial demand thoda sa kam hai. But it is still nutritive. And this plant is very much resistant. Ye absolutely resistant hai to pests. The farmer did not have any problem growing these crops. He had been growing them till harvest time. Whatever was expected, pura harvest milta hai. So not a problem at all. So now the farmer is always bothered about this feature. He wants to change this feature. And this particular feature needs to be changed. So इस plant के लिए ये हो गया undesirable characteristic और इस plant के लिए ये हो गया undesirable characteristic. So now if the stem or the root and the stem and the bud of these two plants can be brought together, chances are there that these two features may be corrected. So what are they doing? They will keep one particular plant attached to the soil. What is this known as? This is known as the stalk. If you see here, the stalk, get a note of these words, use these words in your answer, stalk, and then bring a portion of this and get attached here. Bring a portion of this and get attached here, and then tie them with ropes, or some tissue tapes are also there which they tightly bind around them so that they don't get separated. And what is this known as, which is brought from another plant? This is known as the shore, the part of the stem, bud, root brought from another plant. I am showing here 2, it can be more than 2 also, 2, 3 bhi ho sakta hai. So now when we are talking about the part of the stem which is attached to the soil, usko kya bolenge? Stalk. And part of the stem which is above, that is known as shoon. Then at this region some hormones are also introduced. Some hormones are also introduced. Some hormones are also applied. And then some disinfectants are also used here. Why disinfectants? So that there is no microbial growth, because 2 types of tissues coming together. there should not be any microbial growth. Why hormones? So that the process of cell division can be much faster. Which hormone should be introduced here, injected here? Can you tell me? To initiate the process of cell division? Cytokinin. So cytokinin will be a hormone which will initiate, speed up the process of cell division, and the cells will start fusing, growing much faster. So probably in a period of some 6 months time, we get to see that yes, these are all dried up, falling off, and then now this is a single plant. This has started giving rise to the leaf and the new flowers and the fruit. And when in this new season, when the fruits and everything develop, we get to see that this plant is no more sensitive to pests at all. This new plant is no more sensitive to pests. Rather, it has improved now. It has become resistant to pests. What about the fruit size? This particular property, where the fruit size was very small, and it was not getting any commercial importance, that's why. So that part is also now there. So the fruit is moderate, nutritive hai, the plant is resistant. What does grafting give rise to? Grafting always gives rise to improved variety. Hybrid नहीं लिखना है, improved variety.
मैम वो ये मैंगोज के ना जो प्लांट्स रहते हैं उसमें ऐसे भी करते हैं, देखा मतलब जो उसका ब्रांचेस रहेगा ना, स्ट्रॉन्ग ब्रांचेस रहता है, उसको छीलते हैं थोड़ा सा, फिर उधर ही मड और फर्टिलाइजर्स लाके टेप लाके छोड़ देते हैं, फिर कुछ दिनों में उसको कट करके जमीन में डाल देते हैं तो रूट्स आ जाते हैं,
न्यू रूट्स डेवलप. So that is also another method where improved varieties are made. That method, that is also grafting, क्योंकि वहाँ पे जो when they are slicing off the outer bark, they are putting in the buds of another plant.
नहीं कुछ वो लोग क्या करते हैं, जो ट्री का मतलब ब्रांच है उसको हार्ड कवर रहता है ग्राउंड में, बार्क को वो लोग निकालते हैं और अंदर का जो सॉफ्ट रहता है ना उधर वो लोग मड लगा के टेप लगा के छोड़ देते हैं. हाँ, no, that is because of when we are talking about और प्लांट ऐसा ऐड नहीं करते, बस उसको छील के उसमें मड लगा के छोड़ देते हैं. नहीं, that is generally when we are talking about this, that is only to regrow the same type of a plant in large numbers. But generally, when you are talking about removing the bark, actually the bark is removed to insert, just I have been showing in the other diagram, to insert some other type of a buds of some other plant. That is also another method. Grafting can be done in various ways. But if you are talking about only that, that is only to regrow that similar type of a plant in large numbers. Because that layering type can be
layering, that is get identical copies, not improved copies there. So grafting and vegetative propagation. Now let us talk about some 3-4 major points that you are asked. What are the advantages of grafting? What are the advantages of vegetative propagation? Now, overall, if I ask you what are the advantages of vegetative propagation, why should a farmer go for vegetative propagation? Number one, to get large number of plants in a short period of time. Number two, the farmer is not dependent on any external factors like wind, water, rainfall, nothing. Because these type of plants, you just put them, they will grow generally in large numbers. Third point, now to talk majorly about grafting. Whenever you are asked to write down advantages of vegetative propagation, write 2-3 points specifically about grafting. So grafting mein what is the third point? Grafting is a method by which improved varieties can be obtained. Grafting is a method by which improved varieties can be obtained, and now the farmer can grow this improved variety in large numbers. So when we are talking about the entire concept of vegetative propagation, these are some 2-3 points that you should always remember. Layering, grafting. You are also asked questions between differences between layering and grafting. So that is very clear. Identical, new varieties. And why the new varieties? So that the undesirable characteristics of certain plants can be removed out. A plant can be formed with all good and desirable varieties. So that is the concept about vegetative propagation. ये topic clear है? Doubts है कुछ? 3 methods. The diagram drawing is not there here in this part of the chapter. There is no diagram drawing at all. But differences, advantages, you are asked. Now let us move on to a concept of sexual reproduction. We'll start talking about the sexual reproduction in the flowers first. Sexual reproduction in flowers, as we talk about. The first concept of sexual reproduction, little bit I had discussed the other day when we had started this chapter. The moment we are talking about sexual reproduction, first thing to remember is there has to be a reproductive organ. There has to be a reproductive organ, and this reproductive organ, the cells of the reproductive organ will always undergo a process known as meiosis. If it is meiosis, how many daughter cells are formed from one parent cell? 4. What about the chromosome number? Half of it, whatever the parent cell has. So if it is a concept of sexual reproduction, the first thing to remember is the cells of the reproductive organs will always undergo a process known as meiosis and result in the formation of specialized cells, and these specialized reproductive cells are known as gametes. Talking about the next concept of sexual reproduction is a particular type of a cell. I talk about any cell of a reproductive organ. And then the cell of this reproductive organ is known to have 20 chromosomes. This cell which has got 20 chromosomes, this will undergo the process of meiosis and result in the formation of 4 cells. And each of these 4 cells, if it is the rule of meiosis, by the process of meiosis, it is supposed to have only half the chromosome number, that is 10. Definitely, what are these cells known as? These are known as gametes. So we will write, to write down the chromosome number, we will write this reproductive cell of the organ, that is a reproductive organ. This cell is known to be diploid. What is the meaning of the word diploid? A cell which has got the complete chromosome number. What about this side? I will say this cell is known as haploid. How do we write it? We generally write it this way, meaning this cell has only half of the chromosome number. This cell, known as the gametes, they only have half of the chromosome number, or else we will say it has got only one set of chromosomes. It has got only one set of chromosomes, and it has got 2 half sets or 2 sets of the chromosomes. So what is the simple meaning for you to understand? What is the meaning of haploid and diploid? It has got 1 set or 2 sets. That is what we understand. What was the concept of DNA duplication we studied? What happens in DNA duplication? DNA copying we studied, no? At the start of the chapter, that was the first topic we studied. DNA copying, DNA duplication, क्या होता है उसमें?
मैम वो हमारे अंदर के जो कैरेक्टरिस्टिक्स है तो
नहीं कैरेक्टरिस्टिक्स मत पूछो, नहीं पूछना. Cells DNA duplication मतलब one DNA is getting copied to form another DNA. One chromosome is getting copied to form another chromosome. Here we are saying in each of our body cells, whether it is the cell of the liver, lungs, intestine, or the reproductive organs, testes, ovary, number of chromosome is the same. I talk about this 20 chromosome which is present in every cell of our body, isko bolenge diploid. If it is the cell of the liver, lungs, intestine, तो by mitosis वो 20 के 20 ही maintain रहेगा. But if it is the reproductive organ, then this gets divided to just half, having only one set of the chromosome. इसको क्या बोलेंगे? Haploid. Since we are to talk about the characteristics, since we are to talk about the concept of sexual reproduction, gametes, now we understand. How are gametes formed? By the method of meiosis. What are gametes? Gametes are reproductive cells with haploid chromosome number. What are the characteristics I'm talking about? What are gametes? Yes, gametes are formed by meiosis, that is always understood. Gametes always have haploid number of chromosomes. They always have haploid chromosome number, meaning now you understand what is the meaning of haploid chromosome number, the half of what the parent cell has, what the reproductive cell has. Since they have just half of the chromosome number, what do you think? Should they be able to divide and multiply on their own? Will they be able to divide and multiply on their own? They have just half of the chromosome number. No, they will not be able to divide and multiply on their own. That is the reason they are very much dependent on, or they are dependent on a process known as fertilization and then continue their process of growth. So I would write the third point is not capable of division and growth. So just remember these features. They are not capable of division and growth. They are not capable of division and growth on their own. And we are talking about the concept of fertilization later. So to start with this concept of sexual reproduction, which is the same for the flower also, and for the human also that we study later, these are some of the common features to remember. What are gametes? How are they formed? What are their features? They always have half the chromosome number, and that is the reason they do not have independent existence. They are having a short lifespan. They always have a very short lifespan, few hours to a couple of days only. Few hours to a couple of days only. They can survive. Within this period, if fertilization, then they go ahead form a new organism. No fertilization, they will die out. Clear? Now, after the break, we'll now start understanding the parts of the flower and how reproduction in the flower.
Obviously,
अरे वो ड्रॉप करने का नहीं है
5 मिनट का होते 10 मिनट तक देखती रहना कुछ जाते ही नहीं क्लास के बाहर